Okay, good afternoon. I would like to welcome you on behalf of Pufendorf Institute's Migration Initiative. I'm the coordinator of this interdisciplinary seminar series here on migration. And today's topic is, for some of us, is really beyond our disciplines, but for some of you, probably you're more familiar with. So the theme is the role of genetic geology and archaeology in migration studies, and the ways in which these methods could be used, or what are the challenges in using such methodologies when it comes to studying migrations in the past. And without taking too much time, I would like to just introduce our speakers for today. So Hannes Schroeder is from the Natural History Museum of Denmark, University of Copenhagen. And second speaker, Bettina Bader, and she's from the Institute of European and Oriental Studies and at the Austrian Academy of Sciences. And she's also an archaeologist specialized in ancient Egypt. Okay, now I can leave the floor to you, Hannes.
Thank you very much.
Thanks. Welcome.
Thank you very much for the welcome. Thank you for the introduction. It's great to be here, a little trip up from Copenhagen. The brief was to tell you a little bit about how in archaeology we use isotopes and ancient DNA to look at migrations. I tell you straight away, there's not going to be a lot on isotopes in the talk. I did isotopes in another life, but I'm mainly now doing ancient DNA, and that's what mainly the talk's going to be about. So I've termed this ancient DNA revolution in archaeology. A Swedish colleague of yours, Christian Christiansen, has coined that phrase, another revolution in archaeology, akin to the kind of radiocarbon revolution and whatever we had. And the subtitle is kind of Migration Makes a Comeback. And I think Bettina is going to talk about generally the history of migration research in archaeology. So I'm not going to say too much on that. Just to say straight up that it was not always a popular concept in archaeology. And it has changed over the years. So what I'm going to talk about a little bit, therefore, is a tiny insight into the history of migration as a concept in archaeology. And I'm going to get into how we use now ancient DNA in order to study migration and population movements in archaeology. I'm going to give you a couple of case studies, two if I have time, one if I don't. And I'm going to talk a little bit about the challenges that we face, or the criticisms that we faced as a discipline. I should say I was trained as an archaeologist, but have been for the past 8-9 years or so been working with genetics, so in DNA. So I understand some of the criticisms that are generally coming from colleagues in archaeology. So what do we do? Archaeologists, what they're interested in, generally interested in culture change and generally looking at material culture, and then trying to come up with some sort of explanation as to why that change happened in the past, put very simply. And for at several points in the past, migration was a very popular way of explaining culture change. Particularly, I'd say, in the early days of culture history approaches. So we're talking the archaeology of Gordon Child and those kind of people, where migration was used as the mechanism to explain cultural change. So one group of people moving into another area and somehow kicking in action a chain of events that led to changes in material culture, changes in lifestyle, changes in foodstuff being used, and so on and so forth. Now, this is kind of exemplified by, particularly in the early days, kind of these cultural historical approaches where, here you have a table where different cultures are expanding and retracting, and they have different, kind of material cultures things associated with them. Here the Aegeans starting to build palaces and so on and so forth. And those kind of approaches also kind of culminated in kind of hyper-diffusionist explanations. So you had, people saying that Stonehenge was built by Egyptians, or other even crazier kind of suggestions being made of links between different people. And again, migration was, the way that this should have happened. Then it really fell, these kind of approaches fell out of favor, particularly in the 60s, 70s, kind of the post-processual approaches in archaeology. I don't know if that's a concept that you're familiar with. And I kind of did my undergrad at the tail end of these post-processual approaches, and migration really disappeared from textbooks and from explanations that were inferred. People emphasized much more kind of autochthonous or local developments, so cultural developments, culture change as being the cause of local developments. And all people ignored these types of things altogether and were emphasizing other things like I don't know, how does it feel to walk through a landscape or whatever. One of the kind of classic examples in archaeology of this debate and where migration was also being inferred as one of the kind of possible explanation is the spread of farming into Europe. So here we have a relatively early from the 1965 a publication showing the different dates of farming settlements in Europe, and they start in with the black dots in the Near East, in Turkey, and then moving a little bit later, you have them appearing in the Balkans, and then even later, moving into Central Europe, and then into the British Isles, and so on and so forth. And that span, that process kind of spanned the best part of 10,000 years or so, starting perhaps 12, 13,000 somewhere in the Near East, and then moving into the British Isles and arriving there perhaps, I don't know, 4,000 years before present. And the big question in archaeology for a long time had always been, well, this process, was this down to a movement of people, or was it down to cultural transmissions or a movement of ideas, right? And either, you could support either argument with the archaeological evidence. Now, where kind of isotopes to some extent, and then ancient DNA comes in, is that there are new methods that allow us to study migration or the movement of people by not looking just at material culture, but by looking at the remains of people themselves. So the old saying of pots are not people, that kind of always comes up in this context as well, to say that if you're just looking at material culture, it tends to be very difficult to try and infer whether this was, a culture change was down to a movement of people or to a movement of ideas. But if you analyze the remains of people themselves, you may have a better chance of trying to figure those things out. Isotope analysis are useful in the sense that they kind of relate to a lifetime. So you can establish local or non-local status of an individual. You can thereby infer whether that person migrated during their lifetime. But it doesn't really tell you that much about a population or an ancestral group or kind of those different levels of inference. And this is perhaps where ancient DNA analysis comes in. It can't tell you where an individual necessarily was born, but it relates to his or her ancestry, so the ancestral composition of that individual and the population that he or she belonged to. And it can be used to infer migrations in the past. And if we take this, I'm jumping ahead here. This is kind of how it works in a cartoon kind of fashion, just to show you that. You start off with a bone or a tooth from hopefully a securely dated archaeological context. You extract DNA, you sequence it on some fancy machines. You get sequence data out of it. This is what it looks like. So basically, DNA being composed of 4 letters, A, C, T, and G. And what we're interested in is really the variation between individuals, or in this case, what you generally do at first is to map the different bits of DNA that you sequence to a reference, and you try to see how it varies from that reference. So here, for instance, just to illustrate this, to kind of get an idea of what the data is that we're actually looking at. So here, in this case, the reference that we used is called a human reference genome, and all the bits of DNA that we sequence from this particular bit of bone tend to have a T in that position, whereas the reference has a C. And so that's the kind of data that we're looking at, single nucleotide polymorphisms, as they're called, mutations. You can use other types of data, but that's mainly what we're looking at. And then we're feeding this type of data for one individual or for several individuals into various programs that allow us to infer things like the ancestral composition of that particular individual. We can look at, we can make inferences about demography. So here in this case, for instance, this is a plot that illustrates changes in effective population size over time for, I think in this case, I think these are humans actually, and Neanderthals and Denisovans. There are other things that one can do as well. So here's just a very brief list. I mean, relatively long list. So the first very kind of straightforward thing to do is simply determine the biological sex of an individual. You just have to look at the sex chromosomes. You can look at phenotypic traits if you're interested in those. So eye color and skin color and those kind of things. You can look at kinship patterns, so how people are actually related. Genetics is, of course, the science of relatedness, so you can try to do that. Determining ancestry, and we're going to look a little bit more into that in just a few minutes. Then how that can be used to look at human migrations, demography, local adaptations is another thing. So how humans and environment interacted. Other things that people are getting into now are things like finding evidence for ancient pandemics through pathogen DNA. Other people are not interested in humans at all, but in animals. So you can look at the evolution and spread of domesticated animals, for instance, all those type of things. Here's a kind of, again, cartoonish timeline of the history of ancient DNA. Also, just to give you an idea, so it kind of started in the 80s with this study of a quagga. It's a kind of half horse, half zebra type beast, the first kind of big step in ancient DNA was the invention of PCR. So that's polymerase chain reaction, which allows you to amplify small bits of DNA. But that also, is a problem in a way because you also amplify contaminants. And so some of the first studies looking at mummies, for instance, that we might hear about later on, as well as sensational claims about dinosaur DNA, and this is why I put Jurassic Park here, and getting ancient DNA from amber and so on, probably were all down to contamination as opposed to the real thing. And this is why I call this the Dark Ages, because really almost nothing from this period you can really rely on as a result because of these issues with contamination and so on. But then it's not entirely true. I'm sure there were some very good studies among those as well. But then there, in around 2005, so roughly 10 years ago or so, and simply a technological change happened, which meant that we could sequence DNA in a very different way, in a fundamentally different way, and that could produce a lot more data within a short amount of time. And then you can see that after that, not too long after, a number of very kind of groundbreaking studies happened. So the sequencing of the first full genome of a mammoth, for instance, just 3 years after. First ancient human genome, which we sequenced in Copenhagen in 2010. The Neanderthal genome, which was published in the same year, revealing fascinating facts about ourselves, who we are, and how we relate to our cousins, the Neanderthals. The Denisovan genome, you may have heard about the Denisovans. They are kind of an hitherto unknown hominin species that was just identified by sequencing their genome from this tiny finger bone. The oldest genome to date is about 700,000 years old, and it's from a horse that was found in the Yukon, also done in Copenhagen. And then about a couple of years ago, people started to do really population-level studies. So not just looking at single individuals, but looking at literally hundreds of individuals. And of course, that gives you much more power and in terms of what you can say. And those studies, one of the major themes in those studies was migration. So here's a very kind of, just an overview of what's been happening because of these technological changes in DNA sequencing. It just shows the number of ancient genomes, so human and hominin genomes, that have been sequenced over the last few years. And you can see that in terms of, modern human genomes, so we're talking here about anatomically modern humans, but ancient genomes. We've got over 1,000 by now, and hundreds are being added every year. So this is kind of the situation that we're in at the moment. Like I said, the reason for this is this technological change. It's often referred to as next generation sequencing or high throughput sequencing. It's simply a very powerful way of sequencing DNA, and you can produce roughly so you can sequence one human genome in about an hour. So and that's 3 billion base pairs, and that would have the first human genome that was finished in 2003. It took I want to say a decade, and it costs an incredible amount of money. So now this can be done in no time, essentially. This is just a side slide, just to say that there's a little bit of a bias in terms of where the genomes are from that have been sequenced, ancient human data, mainly in Europe. And this has partly to do with preservation, but also with the fact that in Europe we have the leisure and the time and the money to do this type of research. There are a number also from the Americas. There's only really one, and then a few more from one from East Africa, and a few more from Southern Africa. So that's kind of the picture that we're dealing with here. So let me then get to this question then. How do we infer migrations from genetic data? Now, this is now already a fairly old schematic here showing the world, and showing simply, if you just take the mitochondrial genome, it's its own little genome that lives in the mitochondria, the powerhouses of the cells, and you just look at those at the DNA, you can start reconstructing on a kind of global level the migrations of anatomically modern humans out of Africa, and then how they spread across the globe. And this is migration at its kind of the biggest level, I guess. These letters, incidentally, were completely arbitrary. They just refer to haplogroups, as they're called, so groups of individuals that share the same haplotype, as it's called, so the same motif of DNA, in this case mitochondrial DNA. You can also start to date these migrations here. So based on this concept of a molecular clock, so you can assume a certain mutation rate, the rate at which these genomes change, and then you can try to date these divergence points here. So going from perhaps 200,000 years ago in Africa into Eurasia and Europe, maybe 80,000 years ago or so, into East Asia, and then into the Americas, maybe 20,000 to 15,000 years ago. Now, this is interesting and tells us about kind of the spread of anatomically modern humans across the globe. But archaeologists, we often, we tend to be interested in migrations at a much smaller scale. And the interesting bit is that ancient DNA, as it's been developing over the past 10 years, allows us to do that as well. And here I just want to show you a couple of slides of the kind of underlying principles that make it possible. So one of the first things to note is that if you look at human genetic variation, so just simply genetic variation of humans across the globe, you'll find that it is geographically structured. And this is simply down to the fact that people who live further apart have less chances of mating than people who live closer together. It seems so obvious. And in genetics, it's called isolation by distance. That's the concept that we're dealing with here. So if you look at this, using an actual data set. This is a principal component plot, which was now published almost 10 years ago by now, a decade. And this was really an eye-opener for a lot of people in the field, in the sense that this here shows roughly 1,000 individuals from Europe, modern Europeans, and you can see, and they have been typed at, or sequenced if you want, but typed using a particular technology at about, I think, 600,000 different positions across the genome. And if you plot the variation along two principal components and you reduce it to these two dimensions, you'll find that they tend to cluster in these groups here. So here you have Spaniards and Portuguese people, Italians, the British Isles. And, long story short, the interesting bit is that a genetic map tends to mirror a geographic map of Europe. So there's a close correspondence between geographic and genetic distances, and the same works for other parts of the world too. Now, in terms of ancient DNA, of course, this is interesting because you can start using this, and we still use, these type of reference data sets in order to infer migration. So the first example, really, of this, or one example, is let's see, the Iceman. This genome was sequenced 3-4 years ago, and if you try to plot Ötzi on this genetic map of Europe, you'll find that he actually plots with modern-day Sardinians. Sorry, those of you who don't know Ötzi, I'm sure you all do, but the Iceman lived around 5,000 years ago or so. And the question, now would be, so why does Ötzi plot with Sardinians? Does anybody have an idea? I mean, at face value, if you just look at it like this, I mean, he was found, here's Switzerland. This is Switzerland on the genetic map. Here's Italy. I think is he Italian or is he Swiss? I can never remember. But he was Austrian. Oh, it's Austrian, right? Even worse. So I don't know. But he was, found in this area here. There's actually an Austrian right there, AT. But he plots with Italians rather than with people up there. So, pardon?
Not far from it.
Not far. Yes. Well, it's true. Although this is the interesting bit here is this is mainland Italy, and these are actually they're off on their own from Italians here, and these are not mainland Italians, but they're Sardinians specifically. Yeah.
Especially with Sardinians and Corsicans, so they are very much mixed.
They're very much mixed. Mixed with whom?
Africans and Europeans.
Yeah. Well.
At least Corsicans and so.
There are. I mean, this is what you say is interesting, is also true. But what I should say here, in terms of how these samples were selected, and this is an important point to make, they didn't just sequence anybody that they ran into in the street or tested, but people who could trace their ancestry at least 3 generations back. So we're talking here, about if you had a recent immigrant, of course, they would not necessarily plot on this map. Or if you had admixed individuals, they would also not neatly fit into these clusters, but they would fit somewhere else. Now, but looking at Ötzi again here, so I guess, one of the, what you might think is that, he come from Sardinia? No, but what, that wouldn't make any sense. Why would he? The other explanation is that the genetic landscape of Europe 5,000 years ago looked very different to what it looks like nowadays. But that for some reason it was preserved in Sardinia. And that makes sense. It's an island, it's isolated. There was maybe less admixture over the years. Recent admixture, fair enough, but maybe over the years less admixture, which means that Sardinians kind of still look like what people looked like 5,000 years ago genetically in Europe. Whereas the rest of Europe has changed quite dramatically. So that's kind of the interpretation that we're going with. And it's just to say that genetically at least, the past was a very different country. Now, what a lot of the studies in ancient DNA that we and many colleagues have been conducting, and I'm going to show a number of case studies that were produced by different labs in Europe, mainly in Europe, but also in the States, that kind of try to explain or understand, what kind of processes actually gave rise to this structure that we see today, and why did it change from 5,000 years ago. So if we look then at, again, the principal component plot of Stone Age Europe, so here we're talking 10,000 years ago, roughly, hunter-gatherers, in Europe. Here in gray are the modern reference populations again, modern Europeans. These are populations from the Near East. Here are the British Isles. And these here in the colors, the colored dots are different hunter-gatherer populations. So here we've got Scandinavian hunter-gatherers, so where we are right now, Eastern hunter-gatherers, Western European hunter-gatherers, and so on and so forth. So the first thing to note here when you look at kind of the genetic landscape of Stone Age or hunter-gatherer Europe is that they don't tend to overlap with modern populations. And the second thing to note is that there is some structure among them. So they don't all cluster together, but they show structure between those populations. Now, what happens if we go a bit forward in time, and this here is simply, just to explain that, is a different way of showing the same thing. It's called an admixture plot. So here you have one individual, each line is one individual, then populations, and you have different ancestral components that make up these individuals. Now if we move forward a little bit in time, and we go to the Neolithic, so now we're talking the coming of agriculture in Europe, you see that they tend to plot in this lower part of the PCA plot. So these are early and middle Neolithic populations, and Ötzi plots somewhere. One of these guys is Ötzi. He plots there somewhere, and they tend to here overlap with these populations. You can see them there in the background from Sardinia. Now, what this indicates clearly is that the hunter-gatherers and the later Neolithic populations were not the same people, right? You see that they're genetically quite distinct, suggesting that with the coming of the Neolithic and all these culture changes that we associate with it from the archaeology, were not down to simply a movement or a change in ideas, but involved actually people moving, probably in areas from starting in the Near East. Now another, something that I always ask my students as well, so if these guys originated in the Near East, how come modern Near Easterners don't look like them anymore? And that's simply the same process that we have seen with Ötzi, that their genetics were also replaced in the intervening 5,000-6,000 years or whatever. Now you can play this game as you go further in time. Here we're looking at the Bronze Age, another period in European history that was associated with really drastic kind of cultural changes. Even associated, and this is something that is highly, a bit more controversial, so the links between genetics and language, but it's associated with Indo-European languages, the Bronze Age, that is. And what we see here is basically, if you move on, you have another population here from more Eurasia, called the Amnaya, and what we see is that really there's one little bit missing, but essentially that modern Europeans are essentially a mix of these three, right? You can kind of see modern Europeans here. They tend to be a mix of these three kind of ancestral populations: hunter-gatherers, Neolithic farmers, and then the Bronze Age kind of steppe populations that were coming in from the east. And that's kind of what gave rise to this genetic structure that we see nowadays, plus, I suppose, these isolation by distance mechanisms that I described earlier. So in terms of the archaeology, or what that tells us, I mean, you could argue, well, it's old news in a way, because not only Maragimbutas, but if we go back much further back to the 20s and early 20th century, people were arguing for people moving and bringing in, as she described it, kind of causing major ethnic shifts in the population. Now, the interesting bit is, and this is one thing I would like to bring across, is that we're going to talk about the challenges later, but that ancient DNA, or DNA generally, can not only allow us to infer that a migration happened, but it can do much more. So here is just a very simplistic example of that. It allows us to kind of determine the timing of particular admixture events as well. So here, a simple example of the colonization of the New World. You've got a population there of Native Americans, of course, already existing, Spaniards, in this case, moving in, admixing with the local population. Now what we can do, and this is just a schematic example, but we can pretty precisely date this admixture event within an era by looking at the lengths of these different ancestral tracts in a person's genome. And that's simply because if they're very long, these ancestral tracts, then it relates to a rather recent admixture event. But if they're very short, it relates to an event that lies far in the past, simply due to recombination. So that's one thing that one can do. Another thing that one can do is, in association with the migrations, you can start to look at trying to infer ancient signatures of positive selection, so local adaptation. So how did people adapt due to certain changes in lifestyle, for instance? And the famous example of this is always lactase persistence, so our ability to digest milk after weaning. And here's an example of that, showing that with the coming of agriculture, you suddenly have in different parts of Europe this lactase persistence allele, so the specific allele that conveys lactase persistence, you just have that rising quite significantly, and it's interesting to see that playing out in the genetic data. Another example of stuff that can be done now, not looking at the human DNA, but looking at pathogen DNA. People are also working on trying to infer the spread and evolution of pathogens that traveled with humans, for instance. So here, again, instead of recovering human DNA, you would recover pathogen DNA, and you can study transmission rates and virulence of particular pathogens, plague, for instance. You can see how they're related, and using that, you can then try to reconstruct how a particular pathogen spread across the globe and when, and so on and so forth. Very exciting area of research. Now I'm in the last, how much time do I have? 10 minutes or so? Then I have time to squeeze in a little case study, another case study that's actually going to be published today. So if you're interested, it's a paper that we've been working on for a while, looking at a completely different context, but just I'm going to give you, this as another example of how we use ancient DNA to infer migrations. And here we're looking at the Caribbean Taino, so indigenous inhabitants of the Caribbean. And so what the questions were there that we had was, well, firstly, where did the ancestors of these people originate that first peopled the Caribbean? We also had questions relating whether we could infer aspects of their social organization and demographic history, and I might get into that if I have time. And then, importantly, we wanted to also know how present-day Caribbean populations relate to these to this ancient individual. In the Caribbean, it's a bit tricky to do ancient DNA. It's very hot. It's very DNA doesn't keep very well. But we were able to find a sample out of many that we looked at, and that one was from the island of Eleuthera, just here at the very top of the map in the Bahamas. and it came from a cave. This one, Preacher's Cave it's called, on the north end of Eleuthera. And in the around 2005 and 6, I think, archaeologists, colleagues of ours, excavated several burials in that cave. And we were able to obtain one tooth that we could try to use to reconstruct or to get DNA out of, and it worked surprisingly well for this rather difficult context. And so we managed to get a high coverage genome from this individual, 12.4x coverage. This is very good for an ancient genome. Means we didn't sequence it only once, but 12 times over. And we could use this in order to infer, firstly, the origins of the migration that led to the peopling of the Caribbean, or one of the migrations, perhaps. And so what we can see here on this plot, for instance, is simply a map showing around, I believe it's 50 different present-day Native American groups, and we show the genetic affinity of this genome from the Bahamas to these different groups. And the darker the color, so the more red, the higher the genetic affinity. And what we can see is that they show the greatest affinity with these populations here from the northern Amazon, indicating that the Caribbean was first settled from South America here, or not first, but that the individuals that this, or the population that this individual belonged to, originated in this part of South America. And this tree shows exactly the same, simply showing how these different populations are related in a phylogenetic tree. And here the ancient Taino fits with these Arawakan speakers from northern South America. So that kind of provides one answer to the peopling of the Caribbean. She was female. That's another thing we found out. And then in terms of the demography, this is getting a little bit complicated, but bear with me. What we wanted to know was, if we can say anything about the development of that particular population, how it changed over time. And one thing that you can do is you can look at so-called runs of homozygosity. So you're homozygous in your genome when you got the same copy of DNA from your mom and from your dad. And so if you have long stretches of this, that kind of means that you come from a bit of an inbred population, simply because there's a lot of DNA shared in that gene pool. So if you, and we can use this in order to infer aspects of like past restrictions in population size, for instance. So in this plot here, we can simply, what we plotted these runs of homozygosity here, and we divided them in different bins. So we have very short runs of homozygosity, so short tracts where you have the same copy from mom and dad, and very long ones. And what we see here is that on this end of the very short runs, this is wrong, I should say short ones, sorry. Lots of short runs of homozygosity. They're indicative of ancestral restriction in population size. And this fits very well with what we know of the peopling of the Americas. People in smaller groups probably moving into the Americas. And then these serial founder effects happening again and again with restrictions in population size, and that's exactly what we see here. So you the Taino, which is here in black, that line, it has basically the same trajectory as other types of other Native American populations. But what's perhaps more interesting is the other end. So the long ones, the long runs of homozygosity, they are very low in this particular individual. And we inferred from that basically it shows no evidence of recent inbreeding or isolation. And that's interesting because this individual lived on a very small island, like 500 square meters. So you might be wondering, if you're looking at the Orkney Islands, for instance, nowadays, people there tend to be a little bit inbred, more inbred in the grand scheme of things. But it didn't seem to have been the case here in these islands. And so that kind of indicates that there must have been some mechanism in place where social organization, or social networks or whatever that extended beyond the local scale. Otherwise, they couldn't have, kept these high levels of effective population size, essentially. So lastly, what we looked at with this particular study was this question of how much of Taino ancestry has survived in the Caribbean or in Caribbean populations today. And if you look at textbooks of kind of American history or Caribbean history in particular, the kind of story that you're often faced with is that indigenous populations in the Caribbean in particular basically were driven close to extinction or to extinction within 100 years of the arrival of Europeans, through disease, through resource deprivation, through forest dislocations, through all kinds of different processes. But there are groups in the Caribbean and elsewhere who strongly self-identify with being Taino or being indigenous to the Caribbean, they would have argued for some continuity, so against this myth of extinction. Now, with an ancient genome, what's nice is that you can actually directly test this. So here we have basically a genome with its 22 chromosomes here plotted, and we colored in. Well, this is from a different study, but they colored in different ancestral tracts on the chromosomes, European and Native American and Sub-Saharan African. And on average, modern Puerto Ricans, for instance, have 15% of Native American ancestry, roughly. And the question then had always been, was this ancestry native to the Caribbean, or did it come from elsewhere? There are lots of there's quite a bit of historical evidence showing that Europeans moved people around from South America and from other places into the Caribbean. So with an ancient genome, you can actually try to figure out whether these blue bits here are actually related to the Taino or not. And so that's what we did, and this is what these graphs here represent. So what we found was that the Native American components in modern Puerto Ricans and also other populations are actually very closely related to the ancient Taino, suggesting that there is an element of continuity, so that Taino DNA has really survived through the present day. Okay, so now I'm going to wrap up, and I'm going to just very briefly in the last 2-3 minutes talk a little bit about some of the challenges and critiques that people have been, or that we've been facing, and that on the one hand, there is this idea of essentialism, or ethnic essentialism. So basically, that we can equate a particular cluster that we see or a biological grouping of people, and we give it a certain ethnic label. We call them, I don't know, Mycenaeans, Minoans, or we call them Memnaya, or and there, of course, the danger that we're running into is that we end up with these grand folk narratives that that were so prominent in kind of the 30s and 40s. And there is definitely a problem with that. Another one is what I call reductionism. So, basically coming up with really simplistic narratives based on, a single sample, like in the case of the ancient Taino genome, and also something that archaeologists have been fighting against for a long time, namely these errors on maps, the big migrations that go from one place to another. And lastly, there's sensationalism, so the idea that, for whatever reason, there can be a tendency to exaggerate findings, and also there is kind of an uncontrollable element of, whatever the media do to your study once you've actually published it and what they read out of it. But having said that, I think, and here's just this slide I put in as a just kind of example of the kind of problematic uses, of course, of this type of research, and it can, in aspects anyway, remind you of kind of Gustav Kosinner's, a German archaeologist of the 20s and 30s, settlement archaeology or Siedlungsarchäologie, where he saw a unified Germanic material culture that overlaps with a particular ethnic grouping, and that is linked with particular parts of Europe. And of course, it was, it was used by the Nazis in order to justify their annexations of Poland and other homelands during World War II. Just to qualify this, though, I think the power of ancient DNA is that we can, yes, we make links between biological signals or groupings and particular archaeological cultures, but we can also see how much more complex the story can be, and this is an example of this. So here, this is another archaeological culture that you may or may never have heard of. There's a Beaker culture. It's a particular Neolithic culture, characterized by these very iconic bell-shaped beakers, ceramic pots, essentially. And you find those in various different styles across large parts of Europe, Spain and Italy and into Germany and also in the British Isles. And for a long time, there had again been the question, okay, how do these different groups relate to each other? Are they related? Is it the same group that produces this distinctive pottery? Recently, this is a study that's still in bioRxiv, but they generated data for about 100 bell beaker individuals from different parts of Europe. Here in red, you can see the kind of the sampling locations of those. And what they found was that these bell beaker populations share actually few genetic links, suggesting that there were not one migrating population. So quite the opposite of what we saw in some of the other scenarios there. So here we're talking maybe about some local developments or people adopting different or this particular pottery style because it was en vogue or whatever. And just to make the story a little bit more complicated, and this is, I think, when you have more individuals available and you can look at more samples, you'll find that in the case of Britain, these individuals do actually seem all closely related to each other. And they seem to have, not only that, they seem to have replaced the earlier population. So just to say that it's a, the more samples you get, it can be a very complex story, and using ancient DNA, you can start to try and disentangle it and add more information. So, just to conclude, then, in one slide, I think, whereas this was not possible, 10 years ago, but we're now really at a stage where we can use ancient DNA to identify past migrations. It's possible to tie those movements to particular archaeological cultures, although not language. That's a different issue and a different talk. And then in addition to this, and this is I think why ancient DNA is also so exciting, is because it can provide insights into demography, social organization, for instance, human-environment interactions, and so on and so forth. Okay, so with that, I'd just like to acknowledge some of the sponsors and my colleagues on the Citizen Project, and you for listening. So if you have any questions, please do. Thank you very much.
Yes, I think we have time actually for a few questions. Would you like to take?
I'll take questions, sure.
Yeah. Do you know anything about the origin of the Basques? I always heard that nobody knows where they are from.
Yeah, Basques are also interesting. They also seem to be, and you may have guessed by, and their language does come into it, but they also seem to have, they seem to be quite an ancient population in Europe, and they also tend to be genetically similar to the Sardinians and to those populations. So they seem to be also a remnant of those kind of early farming people that came in.
Hi, thank you. Thank you for the interesting talk. Thousands of questions that come to mind. And the first one is a very general one that is related to some of the things that you've been mentioning on the issue of reflexivity, if you could say so. There is this thought that comes to mind that is coming from José Saramago, the Nobel Prize in Literature, when he says that every dispute about the past is a dispute about the present. How and in which way you, as researchers that deal with this kind of research, reflect upon this, the way what we're presenting here as a material is going to be interpreted. And the second one, which is very related, is by him also, José Saramago. He says that one of the most difficult things for a storyteller when he or she is going to create a narrative is to choose the starting point. And all these narratives that we have related to human movement and so on and so forth, every time that you're going to deal with them, you will be facing this. So my question to you is, to which extent that starting point is always crucial and complicated?
Thanks very much. I should read more Saramago, it sounds like. So with regards to your first question, so this is, it very much depends which context you're working in. I guess, you know, if you're looking at the very distant past, the fact that all of us here in the room have something like between 2 and 4% Neanderthal genetics in or DNA in us, some people care more about this, some less, and then, you know, but if you're getting into areas where, you know, much more recent time periods, so now where I've been working, I mentioned this Caribbean case, and maybe that's what you're specifically referring to. Yeah, these results are to some individuals are of huge importance because they tend to confirm what their grandma had been telling them all along. And so it's important for their own identity, for, you know, how they feel, who they are. You know, individuals I spoke to who self-identify as indigenous Caribbeans, for instance, who, when in school in the US, have been told, no, your ancestors all died out. So you don't have that part of history to go back to. And so therefore, of course, that type of research is very powerful because it you know, allows them to have a link. So in those cases, of course, we do reflect upon it. We do talk to people who are directly affected by the narratives that we put out. But like I said, this is not always the case because sometimes you know, it tends not to affect people as directly always as in this case. Like I said, you know, whether or not you're 2 or 4% Neanderthal, you know, it's interesting for a number of reasons, but it doesn't affect your daily life, really. As for your second question, I'm not quite sure I completely got it. In terms of course, we all, as a point of origin, if you look at it like the slide I showed for mitochondrial DNA, for instance, I mean, if you go way back, there is a unifying kind of message that we all have a common origin. And that's an important one. And that's one that we always tend to emphasize. You know, wherever we live, wherever, you know, whatever creed, whatever religion or political persuasion, we tend to ultimately go back to Africa as the cradle of humankind. And that, you know, that is a kind of fundamental starting point. But apart from that, the tricky part with drawing arrows on a map and with trying to infer migrations is that arrows on a map do tend to have a starting point somewhere on that map, but it's not easy to establish that starting point, not at all. It's not trivial at all. So, you know, again, in terms of human evolution, if you look at that scale, there's a massive debate going on as to, you know, where in Africa we originated. Is it Eastern Africa, Southern Africa, North Africa, with the recent find of 300,000-year-old hominin remains in Morocco. And that's something that we do have to bear in mind, and that's in terms of, you know, if you look at those maps that we're drawing, you know, with steppe people coming into Europe, they do have an origin. But it's one of the weaknesses of having to visualize something in some way in a map, you know, in a map form, for instance. But that's a convenient shorthand, but not necessarily an accurate way of representing what actually happened. So, yeah. Yeah.
Yes.
Cool.
My name is Martin Lemberg Peterson from Aalborg University. I'm sorry to have arrived late at your talk, but I did enjoy very much what I got to hear. I think it's a very fascinating addition to a range of different disciplines, including migration studies, that this kind of newly developed technology allows us to do, because it's a different lens, a more positivistic lens on migration processes. And I think, well, it's just fascinating the way it can take us back. So 2 questions. The first one is about the mixture between the Neolithic agricultural societies and the hunter-gatherers. I don't know if you're aware of the popular science book by Karin Boyce called My European Family, the Last 54,000 Years. But this is popularizing a lot of what you've been talking about. And I suppose this is a question then that you may just say yes, because it confirms what you've been saying. But there's not in her work, it seems as if, or her depiction of science, it seems as if there's not a full replacement. There's not an either or between these societies, actually, hunter-gatherers and fishers and so on, and agricultural communities lived side by side for thousands of years in Scandinavia, and also interbred. I mean, so I think that's very fascinating too. Very different gene pools, different societies, social organizations interacting. So I think, when you go down into the nitty-gritty, messy details of time, you don't see these clashes that sort of come out if you look at it from a very sort of abstract vantage point. So that's the first question, if you agree with this kind of interpretation. The second one is a little bit of a challenge maybe, or a self-reflection, because I think it adds a lot to, for instance, migration studies to be able to backtrace in this way. But in some of also the popular science work on this, there seems to be an assumption that you can jump from this to more wide-ranging statements on politics, development of cultures, social organizations, as you mentioned as well. And quite often I think maybe not, or maybe there's also some limitations. And I was just wondering whether this kind of, do you recognize this sort of deterministic drive to explain everything? Because you didn't have it among the challenges, but maybe you haven't faced it yourself.
Okay, so thank you. As far as the first question is concerned, so I mean, like I said, one of the criticisms or one of the dangers is that you represent very simplistic narratives, right? And so my answer to this would be that it varied a lot from, depends where you are. If we're talking about particularly kind of the Mesolithic, Neolithic transition, there are other, a number of studies actually that show that, what happened in one place doesn't apply to another. I mean, we saw the example of Britain as well with the Beaker phenomenon there. There seems to have been a replacement, but in other areas that doesn't seem to have been the case. There is evidence for the side-by-side that you're talking about in parts of Europe over hundreds of years. There's evidence for admixture as well. The mere fact that we tend to be, a mix of these three nowadays also goes to illustrate that. On the other hand, there is evidence also, as we see here, that actually replacement did take place, so population replacement, and that the interactions were also not always friendly. That there are, particularly towards the end of the Neolithic, there is evidence for intergroup violence, for instance, in the archaeological record. There is associated then also with these massive migrations or population replacements. So we're looking at a pretty relatively big place, Europe, and with lots of different communities, and so I think it would have varied from place to place. As for the second question, in terms of, again, that's a more difficult one for me, and I don't know if I got it quite right, but I suppose, I mean, there are certain things that we can comment on using either isotopes or ancient DNA, and then others where, I wouldn't venture, making comments about. So already commenting on social structure based on a biology, that's already pushing it, quite far. But then you said that these results have some, you mean in the present, that it has certain effects on how people, can you just repeat that part of the question?
Yes, I suppose it's a bit like, in a way, a version of Yaya's question, in that there's increasing interest in this field and in discussing the findings, and you have a range of sort of popular science works that Homo Deus and the others, you know, Homo sapiens, who in a way are repeating a sort of a deterministic explanatory model that we also find in environmental science, for instance, Jared Diamond and so on. And so the question is, can we explain such complex social? I think you already answered it.
Okay, yeah,
So
I get it
guess
now, yeah.
the answer is sometimes maybe we can explain more than in other contexts.
Yeah, I mean, so yeah, some of these elements of trying to, or this tendency of over-interpreting your data is one that's definitely an issue. So I would say it's, you know, one should be aware of the amazing things that it can do, and stay away from things it can't. I mean, yeah.
Okay, we have one last question.
Thanks very much.
Hello, thank you again for a very
Pleasure.
interesting talk. I'm wondering about the map, about the affinities for your case study on the Caribbean. Of course, you don't mean that that particular person carrying that genome came from the Amazon. What you mean, of course, is that both populations come or have close affinity and come from the same place, because those are modern, and that you lack the spatiotemporal depth in this study. Of course, with the challenges that tropical climate has.
Yeah, no, absolutely right. So I in my rushing through it, forgot to mention this is individual that we sequenced the genome from is about 1,000 years old, from the Bahamas. So you're absolutely right in saying that we're inferring here migration by comparing an ancient genome to modern populations, and what we find is that there's a greater affinity between the ancient genome and populations from North and South America, suggesting that the population that the ancient individual came from had shared ancestry with those particular populations. So it's in the sense it's then less likely that her ancestors came from Yucatan, which is just it's 150 kilometers away from Cuba, or even from Florida in the Bahamas. So yeah, you're right.
But of course, you can't exclude the fact if they came from there, but later replacements.
Of course, yeah. So in a way, like what you're saying, like what we had in the European case, that you're saying that these people here who live there nowadays could have moved there in the last 500 years. That's true. But I think here, given that we're looking at only the last 1,000 years, it's unlikely, I think, that these guys here were replaced in the last 1,000 years. But it's a valid argument to make, or a valid point to make.
I usually work with materials that are quite a bit older. So
yeah,
that's where my question comes from.
Yeah.
Thank you.
Yeah.
Are you going to make an intervention?
I just wonder how could you tell the direction? Could it be so that they originated in the Caribbean and then moved to South America?
Also true. There is nothing in terms of directionality that tells you that. There are other types of evidence then. So what we have here actually, and this is where the archaeology comes in, of course. So we know that the Caribbean was probably one of the last parts of the Americas to be occupied by humans, starting perhaps 7,000 years ago or so. That's when the first kind of archaeological evidence starts to appear, together with the Arctic. And this is where people were already established at that point here in these parts. And then there are archaeological. There's also linguistic evidence in this case actually that do seem to point to South America as a potential, place of origin. What I would say, though, is that as we're gonna, hopefully find a few more individuals that will work out, and maybe, a 5,000 or 7,000 year old individual, I'm sure we're gonna find evidence for another link, for instance, from Yucatan or from other places. So it's, it's a complex history, and also this aspect that the point I made about the social organization and that communities were in contact. The Caribbean Sea, this is what my archaeologist colleagues also tell me, was not really a barrier to movement, but people were moving a lot all over the place. But the signal that we got from this individual, it just fits with this idea that that the Caribbean was one of the migrations into the Caribbean originated in that part of South America.
Thank you, Hannes, for this thought-provoking presentation.